Hi, my name is Kinsey Hussein, and I work in the Kerner Biodiversity Lab. The title of my research is identifying growth of rhizobial bacteria from legume root nodules in a controlled environment. So first, what are legumes? Legumes are plants that are a part of the Fabaceae plant family, also known as the Leguminosae. Legume plants produce a pod with seeds inside. These seeds are specifically what are known as legumes. Legumes form symbioses with soil bacteria called rhizobia that are known to fix atmospheric nitrogen into usable forms for plants. They live in nodules on legume roots in return for nutrients from the host legumes. Legumes are often found in fire-dependent ecosystems such as the longleaf pine savanna ecosystem. Legumes are ecologically and biologically important and play a significant role in the upkeep and survival of the LLP ecosystem. They're a reliable food source for wildlife and replenish nitrogen to the soil. The legume-rhizobia symbiosis is significant to the LLP savanna because frequent fires lead to significant loss of nitrogen from the soil. When studying the symbiosis, collected nodules are often dried due to time constraints, and it is unknown if nodules can be rehydrated and still successfully grow after long periods of being dried. This is where my research comes into play. My research aim was focused on whether rehydrated legume root nodules can grow once they are plated, and if so, at what rate. Moving on to how I conducted my research study, root nodules from two legume species were collected. These are known as Galactia regularis and Lespedeza hirta. They were collected from plants in the sandhills of North Carolina in July of 2020 and were dried out on silica beads for 5 months. In early March of 2021, I rehydrated the nodules in preparation of plating the rhizobia. The nodules were surface sterilized and bleached before being crushed and then streaked onto yeast mannitol agar gel plates. The streaking was done with autoclaved wooden toothpicks. You can see images of myself conducting the experiment, more specifically the streaking process. The plates were then sealed and placed into an incubation chamber at 25 degrees Celsius for 3 days before being moved into the refrigerator to halt the growth process. I recorded the data during the growth period on percent coverage and growth of bacteria on each plate. To the right of the methods section, you can see an example of round 1 data collection from the Galactia regularis species over a period of 4 weeks, in which I recorded number of bacterial species and percent coverage for each. This was just the first round of data collection, which lasted approximately 4 weeks. After this, I began round 2 of plating, in which I poured and streaked new plates using bacterial colonies from the plates in round 1. In the second round of plating, the 3 different possible rhizobial species were differentiated by color, pink, white, and yellow, and were all streaked separately. Moving on to the results, as I previously stated, the main purpose of my research was to see if rehydrated nodules have the ability to grow, and if so, at what rate. Before I jump into my conclusion, I'd like to go over the figure below. It is the box plot showing the data collection from round 1 of plating between Galactia regularis in green and Lespedeza hirta in yellow. I'm comparing the percent cover on the y-axis and the time period of data collection on the x-axis. The figure shows the percent cover for each plate over a 3-week period, comparing growth between the 2 legume species. As you can see, there was a continuous growth of bacteria over time, though there was variation in percent cover within legume species across weeks and between 2 legume species as well. In conclusion, the rehydrated nodules were successfully plated and bacteria did indeed grow over time. Rhizobia from rehydrated nodules have the ability to grow on yeast mannitol agar plates. Different bacteria were identified growing on the same plates, and multiple species were common. And it is now evident that we can conclude nodules can be dried for at least 5 months and still successfully grow. I would like to say a special thank you to the Burton family for their generous donation, which allowed us to conduct our research, as well as my faculty mentor, Dr. Kerner and the student mentors that helped me, Alyssa Young and Sarah Gora. Thank you so much.